Fungi cause significant diseases in teleost fish, first recorded by Arderon in 1784. Groups like Phycomycetes, Escomycetes, and Duramycetes vary in impact. Poor water quality, hygiene, injuries, and decomposing organic matter increase fungal infections, primarily affecting external tissues and occasionally internal organs through spore spread. Fungal diseases are common threats in aquaculture, impacting fish health and productivity. Key diseases include saprolegniasis, branchiomycosis, ichthyophoniasis, aspergillomycosis, and shrimpmycosis. Saprolegniasis, caused by Saprolegnia species, presents as white to gray cotton-like growth on fish skin, gills, or eggs. Transmission occurs through direct contact or contaminated water and equipment. Symptoms include skin lesions, lethargy, and opaque eggs, often leading to severe fungal growth. Diagnosis involves observing fungal structures and culturing samples. To manage it, avoid skin injuries, ensure proper nutrition, prevent overcrowding, and disinfect equipment. Proactive measures ensure healthier aquaculture environments and sustainable fish production. Branchiomycosis, or gill rot, is a fungal disease that impacts the gill tissues of many freshwater fish species. Two primary fungal agents are responsible: Branchiomyces sanguinis, which grows in the blood vessels of gill arches, filaments, and lamellae, and Branchiomyces demigrans, found in the parenchymal tissues. These fungi thrive at temperatures between 14 degrees C and 35 degrees C. The disease spreads via waterborne fungal spores that adhere to the gills, germinate, and produce hyphae, which penetrate the epithelium or blood vessels. Clinically, affected fish show weakness, lack of response to stimuli, respiratory distress, necrosis, and red gills from impaired circulation. Diagnosis involves observing clinical signs, examining wet gill preparations under a microscope, and isolating the fungal agent. Control measures include strict sanitation, removing and safely disposing of dead fish, and treating infected ponds with calcium oxide or copper sulfate. Infected fish can be treated using malachite green in specific concentrations. Prevent further spread by avoiding fish transport from infected areas, and regulating feeding rates during warm weather. Effective management ensures healthier fish populations, and reduces disease risks in aquaculture systems. Ichthyophanosis is a fungal disease affecting both freshwater and marine fish. It is marked by rough, granulomate skin and white to gray-white lesions in internal organs. The causative agent is Ichthyophonus hoferi, an obligate parasite with a yellowish-brown, granular cytoplasm. This fungus thrives intercellularly in tissue, with optimal growth at 10 degrees C, appearing 7–10 days post-inoculation. Transmission occurs orally via infected fish or food, through skin abrasions, or damaged gills. Carriers spread infection through fecal discharges, or direct contact between healthy and diseased fish. Early infections may lack visible signs, but advanced stages show granulomate skin, internal lesions resembling tuberculosis, and organ infections causing abdominal distension, exothermia, or swim bladder damage, leading to immobility and death. Curvature of the spine may also occur due to muscle spasms. Diagnosis relies on microscopic and histopathological examination or isolation of the fungi. While there is no treatment, prevention is key. Avoid feeding raw or infected fish, destroy infected or dead fish, and disinfect all tools and equipment. Proactive measures are essential to safeguard aquatic ecosystems from this deadly disease. Aspergillomycosis, caused by Aspergillus species, is an emerging fungal infection in freshwater fish, first identified as a pathogen in farmed fish in 1983. This disease, often linked to stress from pond management, affects cultured species like tilapia. Clinical signs include abdominal distension, darkened skin, lethargy, and fluid-filled body cavities. Histologically, hyphae may be found in the liver, spleen, kidneys, intestines, and swim bladder. Mortality rates can reach 20%, making prevention critical in aquaculture systems. Shrimp mycosis, caused by Loginidium calanectes or Cerulpidium species, leads to rapid tank-wide mortality. These fungi produce hyphae that spread throughout the shrimp's body. Affected larvae display visible hyphae and die within 3–5 days if untreated. Early detection and management are vital to prevent catastrophic losses in